Good evening and welcome to our group's final presentation in Thesis 2. We are Group 10, consisting of Omar Radwan, Khloud, Abdelrahman, Abdelwahab, Mohamed Tarek, and myself, Ahmed Ibrahim. Our topic concerns the detection of coronavirus using machine learning. As an outline for today's presentation, we will provide you with a brief introduction about our topic and the motivation behind its selection. We will also discuss the datasets utilized in our project, the implemented front end, the methodology by which we were able to complete our objective, the generated results, followed by a demonstration that signals our fulfillment of the community deployment's requirements. So to review the current status as of now in May 2021, the international expansion of COVID-19 is unfortunately re-witnessing a steady upwards rise, and it very much remains detrimentally affecting human and economic health in general. Since local area spread takes place secretly, the number of contaminated people has been continually mounting up until today, and that forms a large part of our motivation behind implementing this project. Medical experts today widely agree that precise and early identification of the coronavirus is extremely crucial to diminish the danger of transmission by quickly enforcing quarantining and contact tracing. The perceived value of diagnostic methods depends on the type of test, the time to get the results, the testing accuracy, and the required resources for testing. Our project idea, which we successfully completed, was to use X-ray scans and image detection methods together with the aid of machine learning models to produce a rapid outcome regarding the presence of the disease. A major advantage that we hope to maintain is that our method of detecting the virus will become simpler in design, cheaper in cost, and more usable in comparison to the alternative methods such as polymerase chain reaction or PCR, among others, illustrated in the picture shown. As for our own method, it is a machine learning based approach that focuses on augmenting a pre-trained model to analyze X-ray scans as input samples. We believe it covers all the advantages that any potential method could come to offer, such as being sensitive, have high specificity, is both rapid and easy to use, and also doesn't require a lab for processing. We are also providing this methodology completely free of charge to medical facilities, so that is an added bonus for us. Throughout this final presentation, we will attempt to showcase the proceedings of our very own portal or website, which we have implemented, and explain how it operates by taking in the digital X-ray image, processing it, then segmenting and classifying it, and only afterwards is the classified result returned back to the end user. We will not delve into technical details too much, but still we would appreciate the chance to offer a thorough explanation of the mechanisms by which we were able to create the portal, and that will be explored in the upcoming slides. We will also be showcasing a demonstration of the portal's utility directly after we explain every single stage the image goes through from the very beginning, ever since it's received on the website, up until a result is returned back to the user.
It's Khloud, and we'll start by mentioning how to know if there's COVID-19 in X-ray. This is a pretty classic X-ray for COVID-19 patients. There are some consolidate of bilateral ground glass capacities in the periphery of the lungs, as shown in the figure blue by the yellow arrows. Here it's a normal X-ray, which means it's a negative case as there aren't any of the COVID-19 indications. Now we will be discussing the datasets that were used in this project. The whole world was facing different waves of COVID-19, which rapidly turned to be a pandemic. We started to search for all the related datasets and collect the most updated X-rays daily, weekly, monthly, to be always up to date. The public and private institutions and many universities started to publish various datasets that collected to help in the conducted researches. Regel is the main source of our datasets. We looked for the most credible sources to get our data from, considering that the errors in the medical field aren't acceptable at all. We mainly depend on Kaggle to get our datasets because it's the most credible source for us till now, and the data there is somehow very organized and clear. We usually update the datasets with the newest one after each conducted presentation till this one. Dataset searching techniques. We have used 3 techniques to get our data. First one is conducted researches, consulting previous published researches for the same problem, referring to the mentioned datasets in these articles using the available X-rays that used in the trained model. Second one is Kaggle, using available datasets posted in Kaggle. Third one is campaign. We have started a campaign to collect real X-rays from positive and negative patients and scan them to be used. And once we started the campaign, many people started to help out with their X-rays, and the only concern they had was their privacy. But we managed to solve this by keeping their names and ages unknown. Here we will be discussing the problems that we have faced while collecting our data. We have found many data sets, but the sources weren't credible to be considered. We had no idea about the main source behind those images. Also, the mentioned data sets in the available research papers in the same topic aren't organized enough or clarified, as we have found mixed images for some COVID cases with others with negative COVID cases without separation. Not only that, but most of the available images aren't mentioned to be whether for positive cases or negative cases. But the most common problem was the invalid and corrupted images, and in the upcoming slides, we will be showing some for that. As we mentioned before, the problem in the real data sets that we have collected as hard copies was people privacy, and we respected that while we are using those data sets. Now we will be looking into the collected dataset in depth. Number 1 is a source from Kaggle, and it contains 5,073 samples of COVID-19, normal cases, and other diseases, and it contains organized data for each for its purpose, testing or training. Second one from Kaggle as well, and third, fourth, number 5, number 6, and number 7. Here, some samples for invalid images. We have 4 images, as we can see. They have some arrows with different colors, squares, that can mislead our machine and, of course, affect our accuracy. Here are some samples for valid images, the 2 on the left for COVID-19 samples and the 2 on the right for normal samples. Here's some samples for the real X-rays, left side for COVID-19 and normal side and right side for a normal case.
Moving on to the methodology of our code, the first thing after the image is uploaded to our website, we use Flask, which is the web framework we're using, which is written in Python. Its main job is to link between the image segmentation and classification models. And this is done by passing the image which has been uploaded to the segmentation model and take the result of the segmentation model and pass it to the classification model in order to classify and hand classify whether this image is positive or negative COVID-19. After that, we use Flask in order to print out on the screen whether the X-ray is positive or negative. The image segmentation model and classification model, both are essential for our project. They are going to be discussed further in the upcoming slides. The image segmentation model is very important for us in our model, as it helps the program to identify where the lung is located, and this helps increase our accuracy of the prediction. We decided to use UNet as it helps to identify and segment biomedical images. The UNet has 2 major parts. The first one is the left part, which is the general convolution process, and the right side is the transposed 2D convolution layer, which is done by concatenating the corresponding image from the path in order to get a more accurate prediction. However, before doing all this process, we need to do some pre-processing for the image in order to get it segmented, and this process will be shown in the upcoming slides. In the image segmentation unit, we have used 3x3 convolution layers with a ReLU, and we used max pooling 2x2, and of course the convolution 1x1 layer, which is the final layer. After training the model and testing it, we have reached an accuracy of 92% for the segmentation model. The image shown now is an example of how our program operates in the training part. The picture on the left is the normal chest CT, where from the dataset, which is provided in the training dataset. And the one on its right is the mask of this X-ray, which is also taken from dataset. And the one on the right of the mask is our segmented prediction, which is very similar to the mask. And it's very clear, and it shows that our model is very accurate. And this is how it's done in our training, and this is what we test based on. Now we'll be discussing what happens to the image step by step after it's been uploaded to when it's being sent to the classification model. The first step is that we take the image and read it, and this is how the image is read, as we can see. And this is its original size. We didn't still change the size or done anything to do with the pre-processing. The next step is that we change this image into a grayscale image in case that there's any color inside this chest X-ray. This helps increase our accuracy, as any color can make the program understand something else, and this can cause a decrease in the accuracy. However, most of the chest X-rays are black and white already, so there's no need to change the grayscale. However, this is just a precautionary step in order to make sure that everything goes as smooth as possible. Moving on to the third step, which we resize the image to 128 by 128 in order for it to be passed to the model successfully. We can see that the grid size has changed. It's now 128 by 128, as we can see. The final step in the segmentation part is to make the prediction of the lung. And here's the prediction from the segmentation model itself, with the size being 128 by 128, as we mentioned in the previous slide. Okay, this is one image where we uploaded to our website, and this is the segmented output of it. The full output and results will be shown later in the upcoming slide where we have created a demo. But we can see from the chest X-ray that has been segmented, showing only the lung, which what the program needs in order to increase the accuracy and show the classification where the lung is located and how big is the lung and where is the infected area and so on. Now to the comparison we made based on the models we have used and we have checked online. Our model is the Keras layers, which had an accuracy of 92%. The PyTorch model had an accuracy of 96%. The TensorFlow model had the lowest accuracy of 73%. And finally, the Kaggle code, which is listed in the references, has an accuracy of 89%. We decided to move on with Keras layers, even though PyTorch has the highest accuracy. And that was due to that we were advised to use the same mainframe for both the image segmentation and image classification models. So we decided to use Keras layers for both. And now my colleague Abdelrahman will continue with the classification model.
Now with the classification. The classification part is a sensitive part of our project, as here is where our application classifies and tells whether the lungs scanned are infected or not. So we need to be careful with the model used, as it depends on the accuracy. Our classification is based on convolutional neural network, CNN, that is composed of convolution layers and sometimes dense or fully connected layers. In order to know the best model, we tested 3 different CNNs with the most resources, and they are VG16, Inception v3, and ResNet. During our research throughout thesis 1, we found that classification is not enough and that we always need to increase its accuracy, and that's the job of the segmentation. Our CNN that we chose is VGG16. It consists of convolution layers, max pooling layers, and dense layers. First, the segmented image passes through 3x3 convolution layers of 64 filters. Then it is max pooled. Then it passes through 2 3x3 convolution layers of 128 filters. Then it is max pooled. Then it passes by 3x3 convolution layers with 156 filters, and then it is also max pooled. Then it passes by 3 3x3 convolution layers with 512 filters. Then it is max pooled, and this whole layer is done once again before it goes to a fully connected layer of 4096 twice, and then a fully connected layer of 1000 with an activation softmax. Now with the comparison. The comparison here is between 3 different CNNs, VGG16, Inception v3, and ResNet. First, VGG16 got the highest accuracy of 87%, which was a surprise for us at the beginning. then Inception v3 with an accuracy of 78%, then ResNet with an accuracy of 52%. The results were surprising for us, to be honest, but through research and testing, we knew why this happened, and we are going to discuss it in the next slide in the analysis. But the outcomes of the comparison is that we chose VGG16 over the other two because it is the best in terms of accuracy and implementation. Now with the analysis of the results. First, the VGG16, which is a simple CNN that we did not expect to have the highest accuracy, especially against Inception v3, but we are going to know why this happened. For Inception v3, it is one of the most complicated and most commonly used amongst large data sets such as ImageNet, but shockingly, it got a lower accuracy than VGG16 for two reasons. First, our data set is not as large as ImageNet. This let lose a lot of features during concatenation. That also happens after the concatenation of the segmentation. For ResNet, it is not an algorithm that we preferred, but we tried it as many papers wrote about it, but it came with low accuracy of 52%. We used a pre-trained model, and this was not a good idea for us, as it is hard to maintain, and we could not train it ourselves due to the lack of time. It also appeared that this is used for large data sets.
And it's time for a short demo. Welcome to the COVID-19 detection portal. Please insert your chest X-ray from the bottom choose file. Here we are using some images that left for testing and not used in the training model. The user can just insert their X-ray and click upload. Once we upload, the uploaded X-ray is passing through two stages. Stage 1, segmentation using UNet model. Stage 2, classification using VGG16 model. Basically, as we mentioned before, the X-ray is going to be resized and formatted as an array to be pixeled and go deeper to classify each segment. The segmented image will appear for the user once the process is done. Now here we go, the results appeared. The result for this X-ray is positive with percentage of 94.31%, and that's all. Stay home, stay safe. Now we will be repeating the same process to upload another X-ray, and the result for this X-ray this time is negative with percentage of 92.55%. Thank you.
For the comparison, we are comparing our project with the PCR and the other blood tests. We found that our project is more superior than the other two tests in accuracy, price, time, and accessibility, where the accuracy of our project got around 86% in total, and other tests including PCR got around 70%. We also found that the blood test does not show any effective call for the COVID-19 presence in the body. It just shows that there is an infection and the body is trying to attacking it. One other point is that the price range for the tests, our costs for using our project is for free. People will only need to do chest X-rays in any lab, and it costs less than 500 Egyptian pounds. For example, in Masr scan, it costs around 275 pounds. In Alpha scan, it costs around 350 pounds, and the highest one in price is in Cairo scan, which costs around 425 Egyptian pounds. On the other hand, PCR and blood tests may vary in price from a place to place, and they all have a high price range that can reach up to 2,000 pounds. For example, in 57 PCR test costs around 800 pounds. In AUC drive-through, PCR tests can reach up to 2,000 pounds, varying between AUC students and non-AUC students. For the last 2 points, which is time and accessibility of the tests, our project will be published online for free for the public use, and it can be seen that our project takes less time to detect the presence of coronavirus from the chest X-rays, while on the other hand, Other tests take time to get the results, and they also are not available on a large scale for the people to be done. Moving to the results, we found that the accuracy of our project reaches 86%, and it can be seen that this is a higher percentage than other tests. Also, our project is not affected by the other strains of the virus that affected the result of the PCR test. Since COVID affects the lungs and we use chest x-rays directly to detect the virus, we also reached a result that is better in preserving the time for patients and doctors, which will give a better edge in attacking the virus, and we made it easier and cheaper for the patients to get examined for detecting the presence of the virus. For our community deployment, we reached some doctors, hospitals, labs, and we got different feedback from them. The doctors got inspired with the project and how it could be very helpful in dealing with this pandemic, and they mentioned that there will be a need for a doctor to double check the results from the scanned X-rays. On the other hand, the labs we talked to about our project accepted the idea, but they refused to put it in use for free, which is our main from the beginning. And for the hospitals, they mentioned that we need to get the approval from the Ministry of Health to deploy our product for free, but they aren't accepting. But the Ministry of Health aren't accepting any other means of testing rather than PCR in the meantime. So we put our product online for free to all people, but we're still looking for the legality of our product, as we don't want anyone to take any legal actions against us. We also will put a PDF for how to use our product with frequently asked questions for the people using it, and this PDF was presented to the doctors, labs, and hospitals that we talked to about our project. Thanks. Any questions?